Adaptive defenses are the very specific wing of the immune system, and specifically, they involve the lymphocytes. So the adaptive immune system, the specific defense system, because remember the innate defense system is referred to as the non-specific system, it also can amplify the non-specific portion as well, and it would do this by activating complement as well as the inflammatory response. However, the one shortcoming that does occur is that there has to be an antigen encounter, the antigen from the specific foreign substance, that the lymphocyte has to come in contact with in order to activate it, to prime it. So it takes a little longer. So there's really four major characteristics of the adaptive defenses. The first is that it involves B and T lymphocytes. The second is that it is specific. It recognizes and targets specific antigens. And it's systemic. It's not restricted to just one area, not just to one lymph node, for example, or one organ. And finally, it has memory. So this means that it remembers its attacker. So it remembers that antigen and can form memory, so the next time that it encounters that exact antigen, there's a much more strong or powerful response, and it happens much more quickly. So the two main branches of the adaptive system to know, there's one that deals with B cells, and that's called the humoral antibody mediated immunity. So remember, whenever you hear antibody, you need to think B cell. The second is the cellular mediated or cell mediated immunity, which has to do with T cells. So for the humoral mediated immunity, it involves antibodies, and these are produced by lymphocytes, which circulate in the body fluids. And those antibodies, they bind with specific antigens. So they bind with the target cell, rendering them temporarily inactive, marking them for destruction by other means. Cellular immunity is lymphocytes acting as target cells, and there's a couple different ways in which the cell can be destroyed. Directly or indirectly. Directly by directly killing the infected cell, or indirectly by potentially another lymphocyte or macrophages. But in all cases, remember the cellular immunity is T cells, humoral immunity is B cells. So our next slide is on antigens, and an antigen is a substance that mobilizes the adaptive defenses, and it provokes an immune response. So they're capable of triggering an immune response. Now the word antigen itself, if we break it into two parts, the first part, anta, refers to antibody, and the second part, gen, G-E-N, refers to generating. So the word antigen means that it is an antibody generating substance. And the antigen determines which lymphocyte or cell is activated. And most antigens are large complex molecules that are not normally found in the body. So the immune system sees them as non-self or foreign. Now, antigens can be there's a couple important characteristics about them. First of all, they can be either a complete antigen or an incomplete antigen. An incomplete antigen is called a hapten. Complete means that it has complete immunogenicity, which means that it has the ability to stimulate certain lymphocytes for them to multiply. And also reactivity. So the complete antigen has both immunogenicity and reactivity, whereas the hapten, the incomplete antigen, only has reactivity. The next part of characteristic of an antigen is that it has what are called antigenic determinants, and it can also be a self-antigen. So antigens are present on all the external surfaces of the cell. They're essentially markers that are in the outer membrane for the cell, and they can be anything that's seen as foreign from the body. So if the immune system is trained properly, if those lymphocytes are trained properly, then it's going to be able to differentiate self from non-self. So on this slide, we can see the antigens, and in this case, there's one specific antigen, and there's 3 antibodies that bind to it. And the way that they do this is through antigenic determinants. And the antigenic determinants are the immunogenic part of an antigen. So the part that generates an immune response, essentially. And the antibodies or the lymphocyte receptor, it binds directly to this antigenic determinant, which we can see is shown here. They kind of look like little Legos, so you can imagine them in different shapes. And they work much like an enzyme would with a substrate, so very, very specifically. So one important thing we need to remember is that all of our cells are marked that identify us. So there's something we have called MHC proteins in our membranes. The MHC stands for major histocompatibility complex, and they're self-antigens that identify our own cells, and they're really the basis for things like transfusion reactions, graft reactions between a donor organ and a recipient. And so these MHC proteins are all genetically determined. So it's very unlikely that 2 people will have the same exact MHC proteins. In fact, it's so unlikely that the only case that happens is in identical twins. So lymphocytes now, there's 5 main steps that occur. The first step is the origination, where they begin. And you learned in previous chapters that blood cells develop from the red bone marrow. That's the process of hematopoiesis. So lymphocytes also originate in that red bone marrow. Once they mature in a couple different places depending on their cell. So B cells mature in the bone marrow, and T cells mature in the thymus. So during the maturation process, these 2 things occur. Immunocompetence, they develop their ability to recognize, differentiate self from non-self. Otherwise, if they can't, then they are killed. And also self-tolerance. The third step is seeding. And what this means is that the antigen now, essentially, it's going to colonize and spread out to secondary lymphoid organs. So basically everything that's not either the bone marrow or the thymus. So lymph nodes, spleen, Peyer's patches. The 4th step we'll see in this next diagram here. So the 4th step is the antigen encounter and activation. So when a lymphocyte's antigen receptors bind to the antigen, and the lymphocyte can be activated. Then the 5th step, the last step, is the proliferation and differentiation. So the multiplication of many lymphocytes and the differentiation as well. So our next slide is antigen-presenting cells. And in the case of antigen-presenting cells, there's 3 main types. But antigen-presenting cells, what they're going to do is they're going to eat, think of the Cookie Monster, they're going to eat antigens, they're going to absorb them via phagocytosis, and they're going to present those fragments of the antigens to T cells for recognition. And the most common antigen-presenting cell is the dendritic cell, and it's the most effective antigen presenter that is known. The second type is the macrophage, and finally the B cell. So our next slide is just summarizing what we've already talked about, the differences between the B lymphocytes and the T lymphocytes. This slide is showing the activation and the differentiation of the B cells specifically. And the main thing that happens here is that plasma cells, B cells specifically, they're going to release the antibodies that act against a foreign substance. So this is the clonal selection of a B cell that we've seen. And so if you're a Star Wars fan, you know what the Clone Wars refer to. You know that a clone is basically an army of specific robots that's going to attack. And you can kind of think of these plasma cells as robots that are produced from an original cell. And this is the process that occurs as the lymphocyte is activated. So here, this process, we see the proliferation and differentiation into effector B cells, which are the plasma cells. And I want you to notice that most of the clone cells are going to differentiate into plasma cells. So we have 3 in this example, but we also have some plasma cells, or some clone cells that will become memory B cells. And the memory B cell, what it's going to do is remember what the original response was to the same antigen. So the next time our immune system confronts this antigen, there's going to be an almost instantaneous, almost immediate response to that antigen.